Hello everyone, and welcome to the last lecture of this semester. In this lecture, we'll be covering the nervous and motor systems, and this is chapter 24 in your textbook. The nervous system is a network of neurons that collects information about the external and internal environments, and then processes that information and decides what to do in response. All animals, with the exception of sponges, have some sort of nervous system, but not all nervous systems in all animals are the same. Some are more simple, while others are far more complex. And then the nervous system conveys a lot of different advantages for an animal. One, it allows them to sense their environment. And this is important if you're trying to avoid predators, catch prey, find suitable environments to raise your young or to live, and also to avoid harmful conditions. And so this is really essential for the survival of many animals. As well as the nervous system allows for your brain to function. So as you'll see in a couple more slides, the brain is primarily composed of a lot of different neurons interacting with one another. And so without the nervous system, we would not have a brain, obviously, because it's a major component of the nervous system. The nervous system also warns the body of harmful situations to prevent further injury. So if you are a fish and you get bitten by some sort of predator and it's not instantly lethal, the nervous system will kick in to cause you to run away so you can prevent getting completely eaten. Or if you put your hand on a stove, it immediately tells your brain, hey, this is hot, move the hand, you move your hand away, and then you learn and you remember, don't touch the hot stove, right? So to prevent you from keeping your hand on the stove, so you don't get further injured, and also prevents you from future injury because you remember, hey, hot stove is bad for you, right? And then it also helps to facilitate movement. And this is because the nervous system interacts very closely with your muscular system, your motor system, which we'll talk about that in the second half of this lecture. Neurons are the foundation of the nervous system. And we've talked about neurons in some detail already in the tissue lecture. So if you need a refresher on the structure of neurons, please go back to that lecture because we won't be covering that detail here. But it is important for you to understand the structure and the function of various parts of the neuron in order to understand this lecture. And so neurons generate and conduct electrical impulses. And these electrical impulses are charges, basically positive and negatively charged ions. And when these neurons generate these electrical signals and send them to nearby cells, whether that be another neuron or some other type of cell, this is called firing. And so when you hear me say a neuron is firing, we're generating that electrical signal and transmitting it to some other target cell. Your body has approximately 100 billion to 1 trillion neurons when you're born. And these neurons, for the most part, they will not replicate. This is however many you're born with, is however many you get. And so as you get older, these neurons will start to die off. And because they're not replaced, you end up with fewer and fewer. Now, luckily, 100 billion to 1 trillion is enough to last you a lifetime. If you do things like if you're a heavy drinker or you get into a serious accident or experience brain trauma or something, this will reduce the number of neurons that you have far quicker. But 100 billion to 1 trillion is enough to get you through life. But you will experience a change in how quickly you can respond to things like your reflexes, as well as how easy it is for you to remember things as you get older because you're losing more and more of these neurons, and those neurons aren't firing as well as they used to. The neurons are composed of three major parts of their structure. We have the cell body, and the cell body is kind of the house of the neuron. It's where everything that you normally associate with the cell is located. So the nucleus, the mitochondria, protein production all occurs in the cell body, and it's shown here. The cell body also will interpret the electrical impulses to decide whether it should generate an action potential or not. And you'll see that a little bit later. And then we have dendrites. And dendrites are the part of the neuron that are responsible for receiving signals from nearby neurons or from the environment. And that's shown here circled in blue. And then we also have the axons. And the axon is this part of the neuron. And it's responsible for sending the electrical impulses to the next cell, whatever that target cell may be. And the axons can vary in length. They can be very long, like meters long, or they can be very short. And when you have neurons that are bundled together with connective tissue, this is what we refer to as nerves. So when you hear the term nerves, just think about a whole bunch of neurons grouped together with connective tissue, kind of in this sturdy bundle. And then in addition to neurons, we have our glial cells. We talked about glial cells before as well. These are the support system of neurons. Neurons could not function properly without these glial cells. And glial cells, unlike neurons, are dividing cells. So they will continue to divide so that they can support your neurons and keep them functioning properly. So because these are the dividing cells, a lot of times when you hear people have cancer related to the nervous system, it's because of the uncontrolled division of these glial cells. And so that uncontrolled rapid division of these glial cells can lead to tumors, which can ultimately impact the person's brain and their motor functions and their speech functions, as well as the potential to stay alive. And additionally, the glial cells play a major role in protecting your brain through forming something called the blood-brain barrier. And the blood-brain barrier is a semi-permeable barrier of glial cells that line the vessels that lead to the brain. And this is important because it blocks the entry of a variety of potentially harmful chemicals from getting into your brain, like toxins or metabolic waste or poisons, right? So you don't want just anything getting into your brain, because once you damage those neurons, they're gone, right? So we want to make sure we're protecting our brains. Now, this membrane is not perfect. The blood-brain barrier does allow for small molecules to pass through, and sometimes these small molecules might be toxins or other harmful compounds. And you've experienced this if you've ever drunk alcohol. You know that you feel kind of that lightheaded, inebriated feeling, and that's because small compounds like ethanol can pass through the blood-brain barrier, as well as some minor toxins and drugs will pass past this blood-brain barrier, and we'll talk about a few of those later. And then this blood-brain barrier is very, very sensitive. It's sensitive to hypertension, radiation, and pathogens. And so if it breaks down, it now leaves your brain susceptible to a variety of threats, whether they be chemical threats, it can be threats that seem relatively mundane but are very, very important, like metabolic waste. Some compounds that we produce as metabolic waste are reactive oxygen species, and that can cause damage to the DNA and cells, and that can lead to cancers and various issues like that, or death of cells. And it also leaves your brain susceptible to pathogens. And so we want to make sure that we protect the blood-brain barrier, because it's keeping our brain safe so it can function properly and keep us going. The nervous system is composed of three types of neurons. We have sensory neurons, motor neurons, and interneurons. Sensory neurons are responsible for receiving signals from internal and external environments and relaying those signals to the rest of the nervous system. They often have modified dendrites, which you'll see some of those later on when we talk about the senses. And these modified dendrites often have these specialized receptors that allow them to receive signals from their environment. And these receptors can be chemoreceptors, which receive chemical signals, photoreceptors that respond to light, mechanoreceptors that respond to tactile pressure, like physical pressure and physical changes, baroreceptors that respond to pressure, and thermoreceptors that respond to temperature. There are various other receptors out there as well. There's even receptors to respond to electrical pulses. So there are a variety of sensory neurons out there. Then we have motor neurons, and motor neurons send signals to your muscles and glands, and this is what is responsible for facilitating the contraction of your muscles and the secretion of various mucus and other fluids from your various glands, like your salivary glands and your sweat glands. And they're also the key to the somatic and autonomic nervous systems, which we'll talk about next. And then we have our interneurons. And interneurons are like the middleman between the sensory neurons and the motor neurons. And they make up the brain and spinal cord. The nervous system can be divided into several smaller systems, but all these smaller systems work together to allow an animal to function properly. Our two largest systems are the central nervous system and the peripheral nervous system. The central nervous system, or CNS, consists of the brain and spinal cord, and it's shown here in the diagram. And it's responsible for processing sensory information and deciding what to do. And then we have the peripheral nervous system, or the PNS, which is shown here. And it's responsible for sensing the environmental stimuli and then initiating whatever that motor response will be. And so the central nervous system and peripheral nervous system work together to sense an environmental stimulus, decide what to do, and then do whatever needs to be done. So how this works is we begin with some sort of stimulus. In this example, it's a mosquito landing on your hand. So let's say a mosquito lands on you. As soon as it lands on me, there are neurons, sensory neurons underneath the epidermis that will sense that change in pressure and say, hey, something's landed on me. So the information will be sent through the sensory neuron to interneurons of the spinal cord. And then interneurons of the spinal cord will transmit the information to interneurons in the brain. And then from here, the brain will take the sensory input and decide what to do about it. So it says, hey, there's something that's landed on my skin. What should we do about it? Well, it could be dangerous. Let's get rid of it. Let's use our other hand to slap whatever it is away. And so once the brain has decided what to do, it sends the signals back to the spinal cord interneurons. And then from there, the interneurons of the spinal cord will send the message to motor neurons. And the motor neurons then initiate whatever that response is. In this case, the response would be have my other hand slap the mosquito off of my right hand. So that's how these two systems work together. The peripheral nervous system has several kind of smaller components, well, not smaller components, but can be subdivided into other nervous systems as well. So we have two major motor pathways that are involved in the peripheral nervous system. We have our somatic nervous system, which is connected to our skeletal muscle. So any of those voluntary muscles that you can control that allow you to jump or skip or clap or snap or whatever is all under the control of the somatic nervous system, as well as reflexes. Reflexes are the responsibility of the somatic nervous system as well. And how reflexes work is you have some sort of signal goes from your sensory neurons to the interneurons of the spinal cord, but instead of being transmitted to the brain, those signals are sent directly to motor neurons. And this is important for the survival of animals, especially when you're in danger. If you're outside and you're sitting by a bush and all of a sudden a cougar jumps out, you don't have time for your brain to figure out what to do. You just need to be able to respond as quickly as possible. And so your reflexes allow you to do this because it takes less time for you to respond if the signal doesn't get sent to the brain. And so this is why if you've ever been scared unexpectedly, if you're like me, I'm very flighty. And so I just run away. And then once I'm out of danger, my brain starts to catch up and figure out, okay, what happened? What was that? What just happened to us? So later on, your brain gets filled in. But first, it allows you to respond very quickly because it could be a life or death situation for the animal. Then we have our autonomic nervous system. And the autonomic nervous system, it controls all of the life components that we don't have direct control over. So this includes all the things that are involve smooth muscle, cardiac muscle, and your glands. So digestion and the movement of blood through your vessels and sweating and saliva production. All of this is under the control of the autonomic nervous system. And the autonomic nervous system also can be divided into two systems as well. The sympathetic nervous system, which is responsible for your fight or flight response. And so this is why when you get scared, let's say we just talked about like a cougar jumps out or something, when you're ready to escape, there will be a lot of physiological changes. Blood will be sent from the non-essential parts of your body at that time to the muscles. So blood will leave the digestive tract and things like that and be funneled to the muscles to allow you to run away or fight back. Also, it will increase your heart rate and your breathing rate and dilate vessels in certain areas of your body. So when you're under stress, your fight or flight response is triggered. This is under the sympathetic part of your autonomic nervous system. And then we have a parasympathetic nervous system, which is under the control of most things when you're not in danger. So when you're digesting food or when you're excreting waste and just the kind of normal everyday run-of-the-mill stuff is underneath the umbrella of the parasympathetic autonomic nervous system. So that's just to remember, the sympathetic is when you're kind of scared, you need to escape. It's high stress right? And then when you're not stressed, it's the parasympathetic nervous system. Now that we've talked about the structure of neurons, the different types of neurons, and the different parts of the nervous system, let's talk about how neurons actually work. And so we'll begin with a resting neuron. And a neuron at rest is doing nothing. It's not transmitting signals, it's not receiving signals, it's just waiting for something to do. And when a neuron is at rest, it has a net negative charge in its cytoplasm compared to the interstitial fluid. And so that's what they're showing you in this image down here, that the charge inside the cytoplasm of the neuron is more negative than the charge present in the interstitial fluid. And this difference in charge is called the membrane potential. And without a membrane potential, a neuron will not be able to fire, it will not be able to function. So this difference in charge on a resting neuron is essential to its functioning. And this membrane potential is established due to these proteins on the cytoplasm called sodium-potassium pumps or sodium-potassium exchangers. Either name is fine. And so how these work is these pumps can move sodium and potassium, which are both positive ions, across the membrane of the neuron. But what it does is for every 2 potassiums it brings into the cytoplasm, it removes 3 sodiums. So you're always losing one more positively charged ion and putting it into the environment. And so this makes it so that there are fewer positively charged ions in the cytoplasm and more positively charged ions in the interstitial fluid. And so just to walk you guys through briefly on how this works, you don't have to remember all the various details on how these sodium-potassium exchangers work. I just wanted you to kind of walk you through it so you can understand what's happening. So we have our sodium-potassium pumps, and they bind to 2 potassiums. And when they're open to the cytoplasm, those 2 potassiums will fall out of the pump and be put into the cytoplasm. Once the potassiums leave, that will cause a change in the protein that allows 3 sodiums to bind. And so 3 sodiums will bind the protein, and then when that happens, ATP will add a phosphate to that protein. Once the phosphate is added, the protein will go from being open to the cytoplasm to being open to the interstitial fluid. So it'll flip. And once the protein is open to the interstitial fluid, those 3 sodiums will leave the protein and 2 potassiums will come in. Once 2 potassiums have come in, the phosphate that was added will leave. When the phosphate leaves, that causes the protein to flip again. So it'll go from being open to the interstitial fluid to being open to the cytoplasm. And then we start basically all over. So this is how we put 2 potassiums in for every 3 sodiums that leave. And so before we move on to what happens when a neuron starts to fire, I wanted to remind everybody that the dendrites of neurons are always interacting with various other neurons. So it's not like a chain, like a one-to-one-to-one all the time. What this more likely looks like is, or more closely looks like, is let's say we have a neuron here, we have our many dendrites. And it's surrounded by other neurons. And each neuron will receive signals from various other neuron axons. So there'll be, axons from a neuron here, and here, say here, and let's say here. This is important to remember when we're going into talking about how neurons fire, because each of the axons that from other neurons that are interacting with our core neuron dendrites here, they can send different signals. Some of them may send signals that tell this neuron to fire, while other ones might tell this neuron not to fire. So it's important to remember that it's not like a daisy chain where one neuron is interacting with another, with another, with another all the time. A lot of times one neuron has dendrites that interact with various other neurons in the environment around them. In keeping with this concept, I'm going to add some axons to our core picture up here because I think it will help to understand what's going on in this firing. So we got an axon there, and then we'll have some axons with some nearby neurons here as well. Okay. Okay, so we have some sort of stimulation. Let's say that mosquito comes back for more and lands on your hand again. Now we need to be able to respond. And so a signal starts getting sent through your nervous system, and our core neuron that we're looking at here is in contact with several other neurons. And some of those neurons will send signals to our core neuron that say, don't fire. And that is, in this example, is our orange colored axon. So it will send signals to the dendrites of the neuron that we've been following, and it'll say, don't fire, don't fire. To do this, there are certain chemical signals we'll talk about in a second that are released that will cause ion channels to open up. And if you don't want the neuron to fire, those ion channels will allow for an influx of negative ions, which will keep that membrane potential. But then there are also other neurons that are interacting with our core neuron that send signals to the dendrites that say, hey, you need to fire, you need to fire. And so in this example, they've colored them green. And so when those signals are sent, it will cause ion channels to open up that will allow for positively charged ions to rush in. And remember, if you want to keep a neuron at rest, you want to maintain that difference in charge. If that difference starts to decrease and then the neuron becomes more positive, then we'll start to fire. So what happens is those signals for both fire and don't fire, which are positive and negative ions, will be sent to the cell body. So that's what they're showing you here. So the don't fire signals are coming in, being sent to the cell body. And then the do fire signals, which are positive ions, are being sent to the cell body as well. The cell body will interpret that information and decide what to do. So if there are more positive ions, then the neuron is more likely to fire. And if there are more negative ions, then it will not fire. And so how does the cell body know how many is enough positive in order to fire? Well, there's something called a threshold. And so I've included this image here. You don't have to remember all the terminology in this chart, but I think it's a good, useful way of showing you how this all works. So a neuron at rest, we talked about, is more negative. And so it's just hanging out, it's chilling, and then there's some sort of stimulus. And that stimulus will cause an influx of both these negative and positive ions. The more negative ions that there are in relation to the positive ions will keep the charge negative. But if there are a whole bunch of positive ions, then the charge will start to increase. And then there's a certain amount that there has to be met for the neuron to fire. So if there's a whole bunch of positive ions, then the charge will continue to increase and increase and increase until it gets to something called an action potential. So once the charge is going more and more and more and more and more positive, it will be like, okay, we need to send the signal. And that sending of the signal is the action potential. And so that's when the signal will be sent down the axon to be sent to the target cell. Now our signal is traveling down the axon. And the longer the axon is, the greater the chance that the signal will dissipate before it gets to the very end of the axon, which is the axon terminus. And so to prevent the signal from dissipating, our axons are wrapped in these fatty coatings called myelin sheaths. And the myelin sheaths help to insulate the neuron and maintain the strength of the signal. Between the myelin sheaths, we have these kind of gaps. And the gaps have sodium channels that help to push the signal along, as well as maintain the strength of the signal. So what this looks like is shown in this picture here. So these little puffy white areas are the myelin sheaths. And between those, you can see these various purple proteins in this picture. Now they're a little bit difficult to discern because they're both in purple, but the lighter purple or the pink more colored ones are sodium channels that bring in sodium to the axon. And then the darker purple ones are sodium channels that are pumping sodium out of the axon. So how this works is our action potential will start traveling down the axon, and it will get to these kind of areas between the myelin sheaths. And once it gets there, there's so much positive ions that it will cause these sodium channels to open up to bring in more positively charged sodium ions. So they'll open up and bring sodium in. And this will keep the signal going and pushing it in the right direction and keep it strong. And so it'll keep moving down the axon and it keeps going further and further down. And as the signal is traveling down the axon, once it passes a particular point, there are channels that will remove the sodium from the axon. So that's what they're showing you here. So this at this picture here, the action potential has moved past this region. So we don't need to have those sodium ions present anymore. So there are proteins here that will pump sodium ions back out into the interstitial fluid. And the same with this picture here. So now we've moved past this region. We don't need to have those sodium ions hanging around. So there are pumps that will pump the sodium ions back out into the interstitial fluid. And this is how you keep the signal going in the same one direction. So the action potential will keep chugging along until it reaches the end of the axon. At the axon terminus, there are a lot of different vesicles that contain these chemicals, and the chemicals are called neurotransmitters. And neurotransmitters are these chemicals that are responsible from traveling from the axon of one neuron to the dendrites of another to cause some sort of response. So when we were talking about, we had the orange and green colored axons in our example, and I said that there were signals that would cause ion channels to open, and they would bring either negative or positive ions in. Those signals are these neurotransmitters. And so what happens is the action potential reaches the end of the axon, and it will cause these vesicles to fuse with the membrane. And this membrane that the vesicles are fusing with is referred to as the presynaptic membrane. So once the vesicles bind to the presynaptic membrane, they will release the neurotransmitters into the space between the axon of one neuron and the dendrite of another. This space is called the synaptic cleft. So the neurotransmitters will be released into the synaptic cleft, and they will travel to the postsynaptic membrane, which is on the receiving cell. And on the postsynaptic membrane, there are various proteins that bind those chemicals. And that neurotransmitter will bind to that protein, and that will cause that protein to open up. And that protein being an ion channel, maybe for positive or negative ions. In this case, in this example, it's for positive ions. So that ion channel will open up, allowing positive ions to flood in, which will cause an action potential in the postsynaptic cell. And so what happens then is we don't want that neurotransmitter hanging around in the synaptic cleft, because it will continue to cause the postsynaptic cell to fire. So we want to make sure that we send the signal and then remove the signal, because now the message has been sent on. So what will happen is the neurotransmitters will be degraded by the cell, or they'll be reabsorbed by the presynaptic cell to be recycled or to be reused again. There are hundreds, even thousands of neurotransmitters produced by the body, and they all have different effects on the body. However, we're only going to talk about 4 in this class, and these are 4 of the most well-known, best studied, very common neurotransmitters. These include acetylcholine, which is produced by motor neurons and travels to muscle cells, which causes them to contract. Glutamate, which plays a role in learning and memory. Dopamine, which initiates and coordinates movement, as well as provides people with the feelings of pleasure and happiness. And then we have serotonin, which is an inhibitory neurotransmitter. So instead of causing an influx of positive ions, it causes an influx of negative ions. And it has multiple purposes. It regulates mood, has roles in appetite, anxiety, sleep, healing, etc. And it is the main component of many antidepressants. There are many natural and man-made substances that we can consume that will trick our nervous system. One of these is cocaine. And cocaine gives its users feelings of pleasure because it blocks the reuptake of dopamine by presynaptic cells. So before we kind of dive into the effects of of cocaine, let's do a more mundane example. So let's say that you are eating your favorite food. When you eat your favorite food, you might feel that feeling of happiness. And this is because there are neurons in your brain that are releasing dopamine that cause you to feel happy. So let's say you're eating your favorite food, and when that happens, there are neurons that will release dopamine into the synaptic cleft. And once in the synaptic cleft, dopamine will travel to the postsynaptic cell and bind to receptors on the postsynaptic membrane, and that will cause an influx of positive ions into the postsynaptic cell to result in an action potential. Well, under normal circumstances, once the postsynaptic cell has been stimulated, then the presynaptic cell will reuptake the dopamine so that it can be reused or recycled. But when you're on cocaine, this reuptake does not happen. Instead, cocaine will bind the reuptake sites and prevent the presynaptic cell from reuptaking the dopamine. So instead, the dopamine will hang out in the synaptic cleft, continually stimulating the postsynaptic cell. So this will cause the postsynaptic cell to fire and fire and fire and fire, and it'll just keep firing. And this is why, as long as the cocaine is in the person's system, they will feel that feeling of happiness and pleasure and euphoria. Eventually, your body catches on that something is wrong, and it will remove the dopamine and it will remove the cocaine from the synaptic cleft so that the postsynaptic cell doesn't keep firing. And then, of course, in order to have that same type of stimulation again, you would have to do the drug again. So this is how cocaine is having its effect on the brain. Zoloft and Prozac are called selective serotonin reuptake inhibitors, or SSRIs, and they work in a very similar way to cocaine. But instead of blocking dopamine reuptake, they block the reuptake of serotonin. And so if the presynaptic membrane cannot reuptake serotonin, it will cause the constant stimulation of the postsynaptic cell, which gives the user feelings of contentment or satiation, because that is the effect that serotonin has on the brain. Then we have morphine and heroin. And morphine and heroin do not act in the same way as cocaine and SSRIs. Instead of blocking the reuptake of a natural neurotransmitter, they instead mimic a neurotransmitter. So they mimic a neurotransmitter called endorphins. And endorphins are your body's natural painkillers. If you've ever run long distances or done some sort of endurance training, you've definitely heard of endorphins. What morphine or heroin does is it mimics the endorphins and binds to endorphin receptors in your body, causing your neurons to think, hey, endorphins are here, and cause you to have no pain. Nicotine is an acetylcholine mimic, and it results in the rapid increase of adrenaline, dopamine, and other stimulating neurotransmitters in the brain. Caffeine is an adenosine competitive inhibitor. An adenosine is a metabolite that is naturally occurring in the body that will bind to neurons and inhibit their ability to achieve an action potential. So under normal circumstances, when you're not drinking caffeine, what will happen is this adenosine is being produced throughout the day. So as you're going throughout your day, neurons in your brain, cells in your brain are releasing this adenosine into the interstitial fluid. And the adenosine will bind to receptors on neurons, and it will make those neurons have a more difficult time establishing an action potential. And this is why you feel tired as the day goes on. You have a harder time thinking and focusing because of this. Now, when you consume caffeine, what caffeine will do is it will bind to the adenosine receptors instead. And when it binds, then nothing happens. So the adenosine will be just hanging out in the interstitial fluid, can't bind, and you feel awake. But what you might have noticed is if you don't keep consuming the caffeine once it wears off, you might feel extra tired. And that's because the adenosine is still hanging out in the interstitial fluid because you haven't gone to sleep yet. And so it's just building up in concentration more and more and more. So once that caffeine wears off, the adenosine will bind and then you will feel extra tired. So this adenosine is actually good. I know we like to fight being tired a lot, especially because there's so much to do in modern day. However, these signals is so that your body can go to bed and refresh. When you go to bed, the adenosine is removed, and that's why when you wake up in the morning, you feel refreshed as long as you got enough sleep and you feel ready to go. It's because all that adenosine has been removed from your body and now you feel good. If you don't do that, then that adenosine just hangs around. And once that caffeine wears off, ooh, you're in for it, right? So, ultimately, that's how caffeine is able to make you feel alert. And we have alcohol. And alcohol is a mimic of several neurotransmitters. It has a lot of different impacts on the body, and I've listed a lot of them here. It helps to block glutamate receptors, which makes you feel relaxed. It also slows your reaction time, slurs your speech, and impairs your learning. And then it also blocks dopamine reuptake, similar to what we saw with cocaine, which gives you feelings of pleasure. It stimulates the release of endorphins, which blocks pain. This is why many times when you see someone who's heavily inebriated, they may experience a serious injury, but they don't feel the pain. Now, once the alcohol wears off, that pain is going to be real, but at the initial incident, they may not feel that same amount of pain because the alcohol has stimulated the release of endorphins in their bodies. It also enhances serotonin receptor efficacy, so people feel more content and happy. With all of these compounds and various other ones that I did not mention in this presentation, you do run the risk of addiction and dependency. So some of these, it's very, very apparent. They're hard drugs, so we all know that you can get addicted to them. Or even not so hard drugs like nicotine, we also know is highly addictive. But things like caffeine and alcohol are also highly addictive as well. If you're consuming them over time, or if you've consumed these a lot over time, you're probably very familiar with that, even if it's caffeine. You might notice that if you're a regular coffee drinker and then you skip a day, you might experience headaches, and that's because your body is reacting to the fact that you don't have that caffeine because it's now become dependent. And so this addiction and dependency is difficult for your body as it's adapting to these unnatural conditions, and your brain is like, hey, I want more. It's getting used to, hey, we have these high levels of stimulation. However, you don't necessarily need that stimulation all the time, but you will feel like you need that stimulation due to this increased feeling of pleasure. Now I want to switch gears a little bit and talk a lot more detail about the central nervous system, in particular the brain. And the brain is the kind of the core control room of the body, right? Not all animals have a brain, but a lot of them do. Vast majority of animals do. And the brain exhibits a lot of variability. Between species, there's a lot of variability, as well as between individuals, there's a lot of variability. There's a variety of different neuron structures, as well as neurotransmitters that are produced, as well as the concentration of neurotransmitters that are produced between individuals and between different species. And each species' brain is even morphologically different. So I included some pictures down here that shows you a couple of different brains from different animals, and you can see how there's differences in how smooth the brains are, the size, how much invaginations or kind of like that wrinkly structure. Also, things that you probably can't see from these pictures is the kind of neurons, how many neurons are located in which areas of the brain. That also makes a big difference because certain areas of the brain are responsible for different things. So the forebrain, midbrain, and hindbrain in animals, which we'll talk about in the next couple of slides, are all responsible for different things. And if you have more neurons in certain regions, will dictate the animal's behavior as well as their capacity to learn or to solve problems or their instincts, various things like that. And so all of these things about the brain, these various regions of the brain, we'll talk about in the next couple of slides. And then, of course, the other part of the central nervous system is the spinal cord. And the spinal cord is responsible for relaying the signals from the body at the peripheral nervous system to the brain. And there are regular branchings of axons off of the spinal cord which connect the central nervous system to the peripheral nervous system. As we're going through the various regions of the brain and what they're responsible for, keep in mind that we're referring to the human brain. Some of these details would differ if you're looking at brains from other animals, especially when we're talking about the more abstract elements of the brain like personality and problem solving and intuition. Okay, so the brain has three major regions: forebrain, midbrain, and hindbrain. The forebrain is the largest region of the brain. It makes up about 80 to 85% of the brain mass. And it has various different regions because it's responsible for a lot of different things. So we're going to talk about a few of these here. Just remember some of the general responsibilities. I know it's a lot of information, so it may take a couple of rounds going through this, but just try to remember the different regions and a few things that they're responsible for. So we have like the thalamus, and the thalamus plays roles in vision, hearing, and touch. We have the hypothalamus that plays a role in kind of the more basic necessities of life, including hunger, thirst, sex drive, and homeostasis. And it also controls your pituitary gland and hormone secretions, which we didn't get a chance to talk about hormones or development or reproduction this semester, but the pituitary gland plays an important role in development as well as reproduction. And then also the hypothalamus is responsible for allowing you to feel feelings of pleasure when you do something that's good for your survival or good for the survival of your species. So this is why when you're hungry or thirsty and you finally get something to eat or drink, you feel happy, you feel satiated. Same thing with why there's a lot of enjoyment when you have sex, is because it's good for the species, and so your hypothalamus rewards you for those behaviors. And then we have the limbic system. And the limbic system is responsible for the more the physiological drives and instincts. And there are a lot of different components of the limbic system, but we're only going to really talk about two. We have the hippocampus, which is responsible for long-term memory formation, and their amygdala, which is responsible for associating emotions and sensory input with memories. So this is why when you think of a memory from your past, whether it be positive or negative, you may have certain feelings, or you may remember certain smells. Like if you think of a chocolate chip cookie, you may remember the smell of it or the taste of it, right? It's because of the amygdala has associated some of that sensory input and emotional input with particular memories. And all of these regions of the brain are located at the kind of the central core of the brain. So it's all located in this center region here. And then we have the cerebrum. And the cerebrum is responsible for the more abstract portions of like what it means to be like human. So this is where abstract thought occurs. Language, problem solving, memory, perception, personality, and intelligence are all the responsibility of the cerebrum. And you'll see that they refer to the cerebral cortex in your textbook because the cerebral cortex makes up the vast majority of the cerebrum. Your brain is split up into two hemispheres, the left and right hemisphere, and the left hemisphere is associated with more analytical processes like language, math, science, and things like that. And then the right hemisphere is more associated with abstract concepts like emotions, intuition, and artistic expression. Both the left and right hemispheres of your brain have 4 lobes, and each of those lobes has a particular job. And so we have the frontal lobe, which is primarily responsible for higher thoughts, voluntary muscle movement, and personality. And so people who get tumors in this frontal lobe will also tend to experience changes in their personality because of this function of the frontal lobe, as well as they may experience changes in their ability to control their voluntary muscles because of that's what the responsibility of this lobe is. We also have the temporal lobe, which plays roles in auditory processing, pattern recognition, and language comprehension. The parietal lobe, which it plays a role in sensory interpretation. So this means essentially you take different signals and put it all together and figure out what that all means. So I put an example here, like if you smell smoke, you feel heat, and you see smoke, then your brain takes all of that information in the parietal lobe and says, hey, there's likely a fire. So that's what the job of this lobe is. And then the occipital lobe, which plays a role in vision processing. And I just wanted to throw this out there because one of the like craziest and most aggravating myths out there is that humans only use 10% of our brain. That is not true. We use 100% of our brain. Our body would not waste any time maintaining something that we don't actually use. So we use 100% of our brain. Just keep that in mind. Then we have the midbrain. And the midbrain is part of the brainstem, and it's responsible for transmitting sensory and motor information to and from the forebrain. So the vast majority of sensory and motor neurons pass through this portion of the brain, and it's responsible for deciding what signals are actually important and which are not, because not all signals need to be sent to the forebrain to be analyzed. Some things in your peripheral vision don't need to be analyzed, or things that you might overhear in the background that aren't relevant. So your midbrain decides, okay, is this information important enough to go to the forebrain? Yes or no, and then sends that signal on. And then we have the hindbrain. And the hindbrain is responsible for the more automatic and subconscious processes of your body. And so this is also an expansion of the spinal cord, and it's part of the brain stem as well. And it has three components: the medulla, the pons, and the cerebellum. The medulla and the pons play roles in respiration, heart rate, and digestion, and they're also part of the brain stem. And then the cerebellum plays roles in complex muscle movement and muscle coordination. This is the part of your brain that eventually allows you to do things on autopilot. So let's say you're driving, you've ever been driving and then you don't even realize how you got to another place. It's because you don't have to really think about that driving anymore. You've done it so much that you were kind of doing it on autopilot. Or like me, I crochet. I can crochet without looking or without thinking about it because I'm on autopilot by this point. And when you have damage to the hindbrain, because it's responsible for some of these key automatic processes of your body, like breathing and your heart rate and digestion and muscle control, if you have damage to this portion of your brain, this often can lead to paralysis or death. Now let's talk about the senses. The senses allow you to interpret information about the world around you. Many animals have specialized sensory organs that optimize the sensory neuron's ability to receive signals from the environment. For example, your nose or your taste buds in your mouth or your ears, right? These are sensory organs that help to receive signals from the environment so that your sensory neurons can catch those signals. And the sensory neurons have modified dendrites, which we'll see in the next couple of slides what some of these dendrites look like. And that's how they're able to receive signals from the environment and send action potentials to various other nerves in the, neurons in the nervous system. And then the intensity of the stimulus can vary. And this will also vary the response to that stimulus. So for example, they're showing you here in this graphic that if you have a weak stimulus, it may only stimulate one or a few neurons. And it might also stimulate them very lightly. So they'll only produce a small amount of neurotransmitters. But if you have a strong stimulus, it may stimulate several sensory neurons and stimulate them intensely. So they produce a lot of neurotransmitters so that you can have a more strong and more robust response to those stimuli. And some examples of some senses include sight, smell, taste, sensing pressure, tactile reception, vision, balance, hearing, electroreception. The list goes on and on. We have so many senses, not just for us, but for various other animals as well, that allow us to learn and interpret information about the world around us. We'll begin our exploration into the senses by looking at taste. And taste is facilitated by modified epithelial cells called taste buds. And you can see in this cartoon up here, they're showing you the taste buds. And this is a zoomed in picture of what a cartoon image of a taste bud looks like. And these taste buds on one end have chemoreceptors. And this is the side that faces the mouth. And these chemoreceptors will interact with compounds that are dissolved in the saliva. And on the other end, they are synapsed with neurons. And these neurons will send signals from these chemoreceptor cells to the brain. And so how this works is in the saliva, there are various compounds that are dissolved. And each taste bud can bind to a particular group of compounds. And so, like for example, taste buds that respond to chemicals that are sweet will bind to maybe sucrose and saccharin and glucose, right? And make you feel, will make you register that those particular compounds are sweet. There are also taste buds that respond to compounds that register as salty, sour, bitter, and umami. And so each taste bud can bind to structurally similar compounds. So they're a little bit more versatile than some of the things that we'll see a little bit later on. And so there are a couple different ways that these chemoreceptors can be stimulated. One, they can be stimulated by charges. So there are some charged molecules that are present in the saliva, like sodium and chloride, that come from table salt, NaCl, that will stimulate the influx of positive sodium ions, and you will register that taste as salty or bitter or whatever that is. And so the example they're showing you here shows that the positively charged ions in the saliva, which cause these ion channels to open up and bring those positively charged ions in. And so let's say, for example, this is sodium. This will allow our brains to register, hey, this is salty, right? But that doesn't necessarily how all the cases work. Not all the compounds in food are charged. Many of them actually are not. And so they have to bind to another receptor on the cell surface. And so they're showing you that here. This is a taste receptor that will bind to this particular food molecule. So let's say, for example, that this food molecule is saccharin, which is sweet. And that saccharin will bind to this receptor. And when it binds, that will cause the ion channels to open up to bring in positive ions. And as we talked about before, when positive ions rush in, that could lead to an action potential, which will trigger these taste buds to release neurotransmitters that will travel to the neurons that they're synapsed with and send those signals to the brain that says, hey, this is sweet, right? So this is how this all works. Taste in all animals is not the same. Some animals don't necessarily taste using their mouth or their tongue. Some use a ton of taste receptors on various other parts of the body. So we see this a lot in insects, where they'll have taste receptors on their antenna. So when you see like an ant kind of feeling around and touching things with their antenna, they're basically tasting everything in their environment. We can also see taste receptors on the legs of some organisms, so that as they're walking around or they're grazing their legs against things, they're tasting various things in their environment. And not all foods have just, well, most foods actually don't have just one group of chemicals that it stimulates. There are a variety of chemicals that are present within food that will stimulate various taste buds. So this is why food has complex flavors. So you might eat like, let's say, kettle corn. Kettle corn is salty and sweet. And you will experience both that salty and sweet taste, because taste buds that react to salty things and your taste buds that react to sweet things are both being stimulated and sending signals to the brain, so your brain realizes, hey, this is salty and sweet. And as you're eating, it's not just your taste buds that are involved. You also have sensory cells in your nose that are also involved with your experience of taste. And this is because as you're chewing food, you're producing a lot of volatile chemicals that will travel through the back of your mouth into your nasal cavity. And so part of the ability to taste is your ability to smell. And when you're sick, you might notice that if you're congested, there's a lot of mucus in your nose, you don't really taste food as well. And that's because the chemicals from your food aren't able to bind to the sensory neurons in the nose, which is part of that experience of eating. So because you don't sense the smell of the food, it dulls the taste of your food. So taste and smell go hand in hand. Speaking of smell, smell is facilitated by chemoreceptors as well, but instead of being modified epithelial cells in the mouth, these are neurons, sensory neurons in the nose that have modified dendrites that allow them to bind to very specific chemicals in the air and cause a action potential. So how this works is in the back of your nose, the very, very top, in your nasal cavity, you have these modified dendrites on these sensory neurons that extend into the nasal cavity. And when you inhale, there's lots of chemicals that are suspended in the air. Or when you're eating, right, there's those chemical compounds from your food that are being chewed in your mouth that make it into your nasal cavity. Well, from here, those compounds will bind to the dendrites on the sensory cells, and that will cause an influx of positive ions. So that's what they're showing you in this picture here. So we have the compounds binding to a receptor, chemoreceptor, which will cause an opening of an ion channel. So these positively charged ions will come into the neuron, and that will allow that neuron to have an action potential, which will then, synapse with an interneuron, and so that your brain can interpret what that smell might be. And so this is how we are able to smell different things. But we have to have thousands, hundreds of thousands of these sensory neurons, because each of these sensory neurons can only bind to specific chemical compounds. So unlike the taste buds, where they could bind to a variety of different compounds, the sensory neurons of the nose only bind to one specific type for each neuron. So there are thousands of different types that will recognize all of the different types of chemical compounds that are floating in the air, and that's why you can smell a variety of different things. Sight works a little differently than taste and smell. Instead of using chemoreceptors, we use photoreceptors. And so your ability to be able to see something begins when light enters your eye via the iris. And now once light hits the iris, it will interact with a part of your eye called the pupil. And the pupil will constrict or dilate to regulate how much light enters into the eye. And so once the pupil's decided how much of light is appropriate, the light will pass past the pupil and hit the retina. Now the retina is tissue at the back of your eye that has photoreceptor cells and nervous tissue. And this is what allows you to actually be able to see something. There are two types of photoreceptor cells in your retinas, rods and cones. Rods are light sensitive cells. They're the ones that allow you to be able to see different things at night under dim light. You might not be able to make out what those key details of what those items are in your environment because there's such a small amount of light, and these rods are not really good at discerning details, but you can see certain things. Now, if you wanted to make out key details of those things in your environment, we would need the cones. And cones are also light sensitive cells, but they can make out a lot more detail, and they also can see things in color. They allow us to see in color. The cones are less light sensitive than rods, so in order for your cones to be activated, you have to have a lot more light enter into the eye. Your cones are not activated under low light conditions, like if you're roaming outside at night in moonlight. We have three types of cones: red, green, and blue sensitive cones, and these are what allow you to discern various different colors. And if you have a mutation in any of these cones, it will result in color blindness because you won't be able to detect certain wavelengths of light. So under conditions where things are dark, let's say you're sleeping or something like that, when there's no light stimulation of these rods or cones, there are sodium channels present on the cell membrane that are constantly open. And so sodium is being brought in to these photoreceptor cells, and then that will allow them to send neurotransmitters to neurons downstream that say, hey, don't fire, don't fire. So as long as these sodium channels are open, these photoreceptor cells are producing neurotransmitters that tell the nearby neurons, sensory neurons, not to fire. And because they don't fire, you don't send any signals to the brain via the optic nerve, and so you don't see anything. Now, when light enters into the eye, that changes the circumstances. And so now there will be changes on the receptors of these photoreceptor cells, and they will block the entrance of sodium into the cell. Well, once sodium is no longer able to enter into these photoreceptor cells, then they'll stop producing these inhibitory neurotransmitters. If the inhibitory neurotransmitters are not made, then the sensory neurons will fire. And then the sensory neurons firing will cause the optic nerve neurons to fire, which will send signals to your brain. And this is what allows you to be able to see whatever is occurring in your environment. So one of the key things here to remember is that instead of the stimulus causing an influx of ions, in this case, the stimulus, which is light, is causing a decrease or a cessation of the influx of positive ions so that we don't make inhibitory neurotransmitters, so that signals can be sent to the brain so we can see different things. And there are different types of sight in different animals. We have pretty decent sight as humans, but we don't have the best sight. Some can see really crazy things, like a lot of bees can see UV light, so they can respond to ultraviolet wavelengths of light that we can't see. Some can also see various other pigments that we can't see as well, like infrared. So there are different types of eyesight out there. Also, many animals also have, many insects will have compound eyes, which are almost like having a whole bunch of little eyes on one big eye. So they can make out what's in their environment, almost like a 360, oh, not 360, well, almost a 360 way of seeing what's around them. The ability to hear relies on mechanoreceptors. These are receptors that respond to physical changes like mechanical pressure. And so when you're hearing things, it all begins when sound waves enter into your ear and travel down the ear canal. And then the sound wave eventually hits this tiny membrane, this very thin membrane in your ear called your eardrum. And once the sound wave hits the eardrum, it'll cause the eardrum to vibrate. And then once the eardrum starts to vibrate, it will send vibrations to the tiny bones behind the eardrum, and those tiny bones will start to vibrate. And when those tiny bones vibrate, it will send that vibration to the inner ear. Within the inner ear, we have various cavities that are filled with fluid. And so when the tiny bones send vibrations to the inner ear, that will cause the fluid to move. And also within these cavities, we have these tiny hair cells that look something like this. And when that fluid moves, it will push those hair cells and cause them to bend. Well, when those hair cells bend, it will result in a release of neurotransmitters. And those neurotransmitters will then be sent to the neurons that they're synapsed with and send action potentials down those neurons, and then it'll continue to travel until the signal reaches the brain. And so this is how we're able to hear different things. The louder the noise is, the more vibrations, and then the more vibrations, the more the fluid moves. And so if the fluid's moving more, it'll bend those hair cells more. So the more the hair cells bend, the more neurotransmitters are released, and the louder you perceive that sound to be. On the opposite end, the quieter a sound is, the more subtle the bending of those hair cells. So fewer neurotransmitters are released, and you perceive that sound as being quieter. So these hair cells are very, very sensitive, and they're very fragile. So if you're listening to loud sounds regularly over a long period of time, you might notice that you have hearing issues, or hearing impairment, due to the fact that these hair cells are irreplaceable, and once they're gone, they're just gone. So you want to make sure you're taking care of them and avoiding really loud noises, so that you can maintain your ability to hear over time. And then we also have another very similar mechanism in bats called echolocation. And so what bats will do is they'll send out sound waves that will bounce off of items in their environment and then bounce back to them to be perceived. So for example, a bat will send out a sound wave, which by the way, we can't hear. They're such high frequencies that the most of the human ear, we can't detect them. And so they'll be sent out and they'll bounce off of, let's say, a moth in the bat's environment. And then the sound waves will travel back to the bat. Well, this allows them to interpret those sound waves similar to what we just talked about for our ears, and they'll be able to identify where different prey is or other items in their environment. Touch is one of the more involved of the senses. It involves a variety of different receptors. Mechanoreceptors, thermoreceptors, pain receptors, electroreceptors, the list goes on and on. But in all of these cases, the mechanism is very similar. So each of those types of receptors will respond to or pick up certain environmental stimuli. And once that stimulus is received, that will cause a change in the structure of the cell membrane. And when the cell membrane changes structure, that will cause certain ion channels to open up. And so let's say you touch something that's a little bit warm and you stimulate those thermoreceptors in the skin. That will cause an increase in the amount of positively charged ions that get put into the neuron, which will cause the neuron to fire. And then once the neuron fires, it will send information to the brain. So it's very similar to what we talked about before, but each of these receptors works in a slightly different way to pick up different signals. However, in all cases, you're stimulating either the influx of positive or negative ions to have some sort of effect on those cells to release neurotransmitters so that we can send signals to the brain to respond to whatever that stimulus is. Another concept that I feel is important for us to discuss in this lecture is the motor system, and that's because the motor system and the nervous system are so closely linked. The vast majority of muscles in our body would not be able to contract without some sort of neural stimulation. And so it's important for us to just know some basics about the motor system and how it functions. So before we get into that, I just wanted to do a quick overview again of some muscle tissue that we have in the body. We won't go into extreme detail, so if you need a refresher, go back to the first lecture where we talked about the different tissue types. But we have our cardiac muscle, which we talked about in our circulatory system lecture, which is not under the control of the nervous system. Instead, it's under the control of the sinoatrial node. Then we have our smooth muscle, which is under the control of the autonomic nervous system. And then finally, we have the skeletal muscle, which is what we'll be mostly talking about for the next couple minutes. And the skeletal muscle is under the control of the somatic nervous system. And it's called skeletal because it is attached to your skeleton. And your skeletal muscle is attached to your skeleton or your bones at certain spots using connective tissue called tendons. And so we have those certain spots are called origin points and insertion points. And you can see them in this picture here. The origin is where the muscle starts. So for this picture, the biceps and tricep origins are here. And then the insertion is where the muscle ends. And so you can see the insertion for the bicep and tricep here in this picture. And for each skeletal muscle, they're under the control of one neuron. So there's one neuron per muscle fiber. So there are tens of thousands, millions of neurons because we have so many muscle fibers in our body. And your skeletal muscles are a lot of times organized into antagonistic couplings. So what this means is you'll have muscles on either side of a particular part of your body that will act opposite to one another. So as one of those muscles contracts, the other one will relax, and then vice versa. So you can feel this if you take your arm and put your hand around your arm and flex your arm. When you feel that flexing, you'll feel your bicep will become harder, and that's because it's contracting. But on the opposite end, if you feel your tricep, you'll feel that your tricep feels softer and it feels stretched, and that's because it's relaxed. And then when you release that flexing motion, you'll feel that your tricep now has become harder because it's contracted, and your bicep has become softer because it's relaxed. And so this is that antagonistic effect. When one is contracted, the other is relaxed. When the other is relaxed, one is contracted. And this is what allows us to do things like bend at the elbow and bend at our knees. Muscle fibers are multinucleated cells. We talked about this before. And you can see it in this picture here. They're showing you the muscle fibers, and these darker spots are representing the nuclei. And each of these muscle fibers contains myofibrils. And the myofibrils have many, many sarcomeres. And we talked about sarcomeres a little bit before. These are the contractile units. And the sarcomeres are composed of two types of filaments. We have thin filaments and thick filaments. The thin filaments are called actin, and the thick filaments are called myosin. And so that's what you can see in this part of the image here. This is a sarcomere, and each of those sarcomeres are going to contract simultaneously or relax simultaneously, which allows for your muscle to move as a whole and contract or relax. So the time between the contraction and relaxation of your muscles is called twitch. And we have two types of skeletal muscle, fast twitch and slow twitch. Fast twitch muscle contracts fast, right? That's where it gets its name from. However, it has low endurance. So these are the muscles that you're activating when you do something very quickly before a short period of time, like sprinting or powerlifting. And so these are also the muscles that when you see people getting like really jacked, this is the the fast twitch muscles are growing in size. So they generally will grow in girth and you get that kind of more muscular appearance. And then we have our slow twitch muscles, and these are slower to contract. However, they have more endurance. And so these muscles are good for more longevity type of exercises like long distance running or long distance swimming. And you'll notice that people who generally engage in these more endurance related exercises are not really bulky. And that's because the slow twitch fibers are more associated with lean muscle. So they don't get really big and bulky. Instead, they stay pretty lean. But they do have enhanced amount of capillaries for these muscles and a protein called myoglobin. And myoglobin binds oxygen and allows these cells to have more oxygen. And this is important when you're trying to do a particular activity for a long period of time. You need lots of oxygen to be feeding those muscles so they can keep doing that activity for a long period of time. So just remember, fast twitch muscles, muscle fibers are for very fast reactions over a short period of time, whereas slow twitch muscle fibers are for more low impact activities over a longer period of time. You may recognize the picture in the upper left corner, and this is because this is the same picture that we had when we first talked about skeletal muscle, and we briefly talked about contraction and relaxation. And I included it here, that way you can orient yourself to what's happening in the bigger picture as we go through the stages of muscle contraction. And so just as a reminder, here is an example of a relaxed sarcomere where the filaments are further apart. And then we have our contracted sarcomere where the filaments are closer together. And then the blue are the actin filaments and the red are the myosin filaments. And the myosin have two components, the myosin heads, which are the more circle portion of this picture, and then the myosin tails, which connect the heads on both sides of the sarcomere to one another. And so if you want to relate what those structures are to this center picture here, this here is the myosin head. This is the myosin tail. And then this is the actin. So as we're going through and talking about muscle contraction, that way you can relate this image to what you see in the top left picture there. Okay, so we're going to begin talking about muscle contraction actually at this stage here, where the myosin head is attached to actin. And so in order for a muscle to contract, the myosin has to bind to actin several times and pull the actin filaments closer and closer together. So first, if we're already attached, the myosin head needs to detach from the actin. So in order to detach the myosin from the actin, ATP will bind to the myosin head, and this will change the structure of the myosin and it will release the actin. So now that the actin is released in the detachment stage, we have to have the reach stage. And the reach stage is to get the myosin head ready to reattach again. And so for this to happen, the ATP turns into ADP and phosphate. When this happens, it releases energy. And that energy is used by the myosin head to change its position. So instead of being kind of more contracted or kind of more in a stronger angle, I guess, if you will, it kind of bends, and this is why they call it the reach. It'll bend so that it can bind to the actin further downstream. And for it to do this, it has to have a lot of energy. It builds up a lot of potential energy. So the best example I could think of is, think about a rubber band. If you have a rubber band and you're just holding it, there's not a lot of potential energy. But if you take that rubber band and pull it, now there's a lot of energy built up in that rubber band, so that if you release it, it'll snap back or it'll fly or do whatever. So that buildup of potential energy is in that rubber band is very similar to the buildup of potential energy in the myosin filament. It's bent back, and in that bent back position, it has a lot more energy to reattach to actin. And so that's exactly what happens. The phosphate is removed from the myosin head, and it will allow for the release of that energy so the myosin can reattach to the actin. And so that's what you see here. And so now that the myosin is reattached, that's great, but we still haven't gotten a contraction yet. We don't get that until the ADP is removed, and then we get the step called the pullback. And so when the ADP removes, that causes a change in the myosin head, which will pull the actin in the opposite direction. And so this process happens over and over and over again. And there are many myosin pulling on a single actin filament. So if all the myosin heads, which you can see in this picture up here, all these myosin heads are pulling on the actin together, they're detaching, reaching, retaching, pulling, detaching, reaching, reattaching, and pulling. They keep doing that. All of them are doing it at the same time. It'll bring those actin filaments closer and closer and closer together so you can get a contracted sarcomere. Now, once your muscle is ready to relax, this process will stop. There will be ATP will bind to your myosin, and they will release, all of them will release the actin, and the actin filaments will naturally snap back to their position where they're further apart from one another. And so this will allow your muscle to relax. And then once you're ready to have your muscles contract again, then a signal will be sent from your motor neurons, will send neurotransmitters to the muscle fibers that will cause the ATP to turn to ADP, and it starts the whole process over again to get that contraction. The last thing I want to mention briefly is the skeletal system. And I only want to mention the skeletal system because it relates to your motor system. This is the site of muscle attachment, and you wouldn't be able to move as effectively without your skeleton. And so the skeletal system provides shape, structure, and support to an animal, as well as a location for muscle attachment. It allows for anchoring of your organs so they don't kind of just migrate throughout your body, which would be pretty bad. And also provides protection for the various soft organs like your skull, your rib cage, and your vertebral column. Protects various soft and vital organs of your body. They're also a site where red blood cells and white blood cells are produced, which we talked about in our circulation lecture. And they're also a mineral reservoir. There are a lot of minerals that are required for every cell in your body to function, including calcium. And so if you are deficient in calcium or you're low in calcium, your bones can act as a reservoir for the calcium for your cells to get the calcium they need. There are 3 types of skeletal systems. We'll just go through these quickly. We have the hydrostatic skeleton, which is present in a lot more simple animals like jellyfish. This is a fluid-filled body cavity. There are no bones or anything involved here or cartilage. It's just a lot of fluid that's kept under high pressure. And this provides shape and structure to the animal as well. Some simple animals like jellyfish, squid, and worms have a hydrostatic skeleton. Then we have an exoskeleton, which we briefly talked about in our animal diversity lecture. And this is a rigid outer coating. So instead of having a skeleton on the inside, it's almost like they have the skeleton on the outside. So they have this coating made of chitin that protects them from the environment and from threats. And some animals that have these are insects, crustaceans, turtles, and clams. And then finally, we have an endoskeleton, which is what we're most likely familiar with because that's what we have. We have internal mineralized tissues. In a lot of vertebrates, we have bones, but also we talked about cartilage and cartilaginous fish. And we also have joints between the bones that allow for us to have some flexibility. For some of these joints, like the joints of your elbow or the joints in your fingers, but some of these joints are also not very flexible, like the joints between the bones in your skull. You don't want those moving around. So not all joints are flexible, but joints are just where 2 bones meet. And some examples of animals that have an endoskeleton include humans, fish, amphibians, birds, sea urchins, and it goes on and on. There are various illnesses and disorders that are associated with the nervous system, the skeletal system, and the motor system. And so I've listed a few here. Some of these we've already talked about in previous lectures, or we've talked about in this lecture already, but I just kind of go through a few just to give you some idea of what can happen when things go wrong. So we have heritable sensory autonomic neuropathy, And this is a genetic disorder that makes an individual unable to feel pain. And this is very dangerous because the ability to feel pain allows you to remove yourself from whatever dangerous conditions that are in your environment. So imagine if you had your hand on a stove, but you couldn't feel it. This would be very detrimental because even though you can't feel the pain, your tissues are still being destroyed by that heat. So if you can't feel it, feel different pain, it increases your chance of sustaining severe injury. So it's a very dangerous disorder. We always talked about brain tumors and how brain tumors are a result of uncontrolled replication of glial cells in the brain, and how this can result in changes in personality, loss of motor skills, and even loss of certain sensory abilities. Multiple sclerosis we talked about in our immune system lecture, where this is a targeting of the myelin sheaths on the neurons of the brain, and it can result in a gradual loss of motor control, balance, coordination, and control over various bodily functions such as excretion. We have Parkinson's disease, which is a result of a loss of dopamine production in the neurons of the brain. And this can lead to the characteristic tremors and slow movements that we see in people who have Parkinson's. It also can impair the person's posture and balance. They lose their ability to speak. And then they also lose their ability to write. So it becomes a very dire situation where you're almost kind of trapped in your own body, if you will. We've talked about color blindness, where you have non-functioning cone cells in your retinas. This is actually more common in men than it is in women, but it does occur in both genders, both sexes. And we have osteoarthritis, which is inflammation and pain of the joints and breakdown of connective tissue, so the cartilage between where the bones meet. That cartilage gets broken down, which we talked about in our immune system lecture. And then osteoporosis. So I mentioned before that your bones are a reservoir for essential minerals. If your body's not getting enough calcium, it will start to take from your bones, and this will lead to your bones becoming more brittle and have like holes in it. Osteoporosis really just means bones with holes, and this can be very detrimental because that leaves you prone to breaks and falls, which can cause serious injury to a person. And this is more common in women, particularly after menopause, as your body has a harder time getting the minerals it needs. So these are just a few illnesses and disorders that are associated with the nervous system, the motor system, and the skeletal system. Yay! You made it through the last lecture of the semester. Congratulations. It was a long ride, I know. And especially this lecture, it was very dense. I know the nervous system can be a bit complex, so please watch this lecture a couple times, as many times as you need, so that you can digest the material appropriately. Go ahead and read chapter 24, sections 1 through 16, and then 19 through 21. When you're going through the sections that talk about the brain, just focus on the more general, big picture things. There's a lot of detail there. I just wanted you to make sure you know the different regions of the brain and what they're responsible for. You don't have to necessarily go into as much detail as they do in those sections. And then go ahead and do the chapter 24 learning curve assignment. And I've included a lot of videos here as well that will help walk you through the various concepts we talked about today. In particular, some of the really good ones are how the senses work. They're a little bit more detailed than what we've gone over in this lecture, and you don't need to know all of those details from that video. But as they're going through and walking you through how the senses work, just keep in mind the key details we talked about in this lecture, because I think they do a really good job of explaining those things and connecting all those concepts. And then also some videos here on how muscles contract, as well as how neurons fire. So if you need some more visual representations of the concepts that we talked about here today, please watch these videos. I think they're really helpful at explaining these concepts and these processes. All right, excellent. You made it through the last lecture, and then next you'll have exam 4, and then of course if you're wanting to do the final exam, that will be made available to you soon as well. Alrighty, congratulations and have a good day.